Hi everyone. So today in this video we will be discussing about the single cell technology and how it has its various application in pharmaceutical and biotechnology industry. And so this technique is quite useful, and its understanding it involves the understanding of cellular heterogeneity and biological process at various level. And this powerful approach allows us to examine individual cells within complex tissues and populations, and that reveals even insights that were previously masked by like bulk analysis method, for example by using bulk RNA-seq data. Well, at its core, this single cell technology, it involves a range of techniques that isolate, analyze, and characterize individual cells, minded at a single cell level. And the most prominent of these is single cell RNA sequencing, or or we also refer it as scRNA-seq. And it provides a comprehensive view of the gene expression profile in individual cells. And the process begins with cell isolation, because in general, whichever cell you take, if it is that individual cell, there are like 20 to 30, 20 to 25,000 of genes set, and you can get its expression level in each of the cells. And often using even a microfluidic device or flow cytometry. And once isolated, the cells, here you can see in the diagram, the cells undergo lysis, and their RNA is captured and converted into complementary DNA, which we call as cDNA. And this genetic material is then sequenced, that produce a vast amount of data that requires sophisticated computational analysis. And in pharmaceutical and biotech technology industry, the single cell technology has found various applications, transforming the drug discovery, and also it is utilized in developmental process. And one of the most significant application is in oncology. Cancer is like heterogeneous disease, and the single cell analysis allows us to dissect tumor complexity at an unprecedented level. And by examining the individual cancer cell, we can identify a rare cell population that may be responsible for resistance or even metastasis. And this information is quite crucial for developing more effective target therapies, and to understand treatment resistance mechanism. Well, in the field of immunology, the single cell technology has been instrumental in unveiling the complexity of immune cells, and it has enabled the identification of new cell subsets, and that has provided even insights into how immune cells respond to various stimuli, including pathogens and therapeutic interventions. And this knowledge is quite important for developing more effective vaccines or even immune therapies. Well, this single cell analysis is also important to understand. For example, it has also been implicated even in the neurodegenerative diseases. I will just show you one example how we can analyze the data through one of the aging mouse data. And by examining the individual neurons and glia cells, and we can even track disease progression at cellular level, and that can potentially lead to early diagnosis and more rapid treatment interventions, including for Alzheimer's or even Parkinson's disease. And in case of drug discovery, single cell technology is enhancing the development of personalized medicines by analyzing how individual cells respond to particular drug, and that can be even utilized. That data can be utilized to predict treatment efficacy and potential side effects with certain accuracy. Well, this approach is particularly valuable in identifying which patients are more likely to benefit from specific therapies, potentially reducing the cost and time of clinical trial. Well, the technology is also providing invaluable. It has been found to be invaluable in the development of cell therapies like CAR-T cell therapy for cancer, or even single cell analysis, that it allows for selection of most potent and specific immune cells for engineering and improving the efficacy and safety of this treatment. Well, now I will show you one example how you can so here I am going to show you one example. So here, this is one of the website where most of the single cell data you can find, like the single cell portal. And here I have selected one of the data which is of aging mouse brain. And here you can see there are almost around 37,069 cells, single cells. And this is kind of atlas of aging mouse brain, a single cell transcriptomic atlas of the aging mouse brain, and that reveals coordinate and cell type. So this is one of the data which has been published in Nature Neuroscience in 2019 paper. So after going to this website, you can easily examine the Tishney plot or even the cell type specific plot, and how you can identify like cell class by aging, or all cells by aging, and many different types. So for example, if you want to see, so for example, there is one housekeeping gene, for example, gap. Let's identify the gap DH. So you can easily check the gap DH. So you will see like there are many different cell types, and it is mostly abundantly present in most of the major cell types, including all the cells, even in the oligodendrocyte, OPC, oligoprogenitor cells, and progenitor cells, and even neuron cells, macrophage. So you can easily see their like distribution. And if you want to see its quantitative like distribution in terms of violin plot, you can also see the violin plot graph, like its cell type specific distribution. And if you want to see its suppose if you want to see its correlation with certain other protein, for example, let's see with the tubulin like beta 3 tubulin TUBB3. So this is, if you don't find any sort of genes, you can easily check the gene card where you can find. So here you can see this is a correlation graph, and this also gives you the Spearman constant rho value, and it is quite positive, and this shows that these two genes are positively correlated, although it is not too high, but they are still positively correlated. So even you can even do the filter out. For example, you can filter if you only want to see in certain cell types. For example, in the neuron, then you can omit all of this. If you want to see in only oligodendroglia cells, or depending on the age, like 21 to 22 months, or even 2 to 3 months. So depending on that, you can filter out those cells. Or if you, depending on the cell class, you can even filter out those cells which you don't want here. And again, if you want to see the dot plot, for example, multiple cell type specific dot plot structure, and you can examine multiple, for example, let me see, map2. Map2 is one of the marker for the protein, which is widely found in the hippocampus neuron cells. And so you can easily find that these neuron cells, they have like high expression. So this red color shows the highest expression, and the blue color shows the zero expression. So this is like a spectrum, and you can even check the heat map. So you will find like individual this subject, and individual uh, sample based expression. So this shows that different proteins. So this is how you can easily examine it, and you can plot, and you can examine the expression of certain genes. So this tells you the cell type specific expression of any particular target genes. So if you have any question regarding the single cell technology and single cell based analysis, you can always ask me, or even you can put your question in the comment. I'll get back to you. Thank you.